Epigenetics by Katie Magali. What is epigenetics? Epigenetics is the study of changes in organisms caused by modification of gene expression rather than alteration of the genetic code itself. Epigenetics goes hand in hand with phenotype plasticity, which is the idea that the genotype creates a platform for possible phenotypes, and that the environment around you holds the instructions for triggering those phenotypes. Your environment can give instructions for timing of gene expression, the location of the gene expressed, as well as the amount being produced. What mechanisms control epigenetics? So histones are large proteins that the DNA wind around. Methyl groups and acetyl groups attach to these histones and create modifications to either loosen or tighten the grip DNA has on these histones. With the addition of an acetyl group, the DNA will loosen its grip on the histones and genes will be easily expressed. With the addition of a methyl group, the DNA tightens and prevents expression of certain genes. DNA methylation is carried out by methyltransferase, which is an enzyme modification adding methyl groups to cysteines and can situate them into major or minor grooves of the DNA, affecting protein interactions. What causes epigenetic changes and what does this mean for us? With consideration to our genome, the ability for our genes to be expressed is all up to external influences. This means that we are not helpless to our genes, and our lifestyle choices and chosen environment are what really determine how our bodies will behave. Some of the things that change our gene expression are toxins in our environment, drugs, aging, and diet. To show you the reality of what our environment does to our bodies, we look at two sets of monozygotic identical twins. On the left, we see a chromosome from each twin. Yellow shows overlap in gene expression, while red and green show various differences in the phenotype based on hypo or hypermethylation. Hypomethylation is the loss of a methyl group. Hypermethylation is the addition of a methyl group to the DNA. In the 3-year-olds, we see that there is a lot of yellow, which shows epigenetic tags are in the same place. At 3 years old, these twins seem to be developing in the same ways in relatively similar environments. Looking at the 50-year-old twins, we can see there is lots of red and green, showing differences in where the epigenetic tags are. This makes absolute sense. These 50-year-old twins grew up similarly and most likely exhibited similar epigenetic tags when they were younger. But as they developed, their environments changed, aka the food they ate changed, their jobs changed, their partners changed, and this all leads to differences in gene expression. DNA hypomethylation is almost a constant companion with hypermethylation of the genome in cancer cells. Just usually, they are targeting different sequences. This diagram shows a transcribed active tumor suppressor gene. When DNA is methylated in the promoter region of the genes, where transcription starts, genes are inactivated and silenced, which means in cancer, epigenetic silencing through methylation is fairly common and leads to the silencing of normal tumor suppressor function. DNA hypomethylation was the initial epigenetic abnormality recognized in human tumors. It affects repeated sequences and is usually linked to the progression of cancer cells. Nutrition and epigenetics. Nutritional epigenetics is the ways in which food affect the pattern of gene expression within our bodies. The foods we eat instruct our body to extract certain nutrients, and diet is easily studied. There are foods that can control DNA methylation. If your body is not doing its share of methylation, doctors can spot the problem with increased homocysteine, the amino acid byproduct. Methyl-related nutrients include folate, methionine, vitamin B12, and vitamin B6, and have been linked to decreased risk in breast cancer, colon cancer, and pancreatic cancer. They have found that drinking even moderate amounts of alcohol may prevent your body from properly absorbing methyl-related nutrients. Nutritional epigenetics is the way in which food affects the pattern of gene expression within our bodies, as we've covered. The foods we eat instruct our body to extract certain nutrients and use them for energy. When we eat highly methylated foods like folic acid, vitamin B6 and B12, methionine, and choline, we add methyl groups to our DNA. Diet is easily studied. In an experiment, these mice were genetically identical. We can see their individual responses to the agouti gene are very different. The agouti gene may be methylated, leading to a thin, brown, healthy mouse, or demethylated, leading to an obese, yellow-furred mouse. This is controlled by their particular diets. One was given highly methylized diet, while the other lacked those nutrients. These mice were then impregnated and the demethylated yellow mouse was supplemented with a diet rich in methyl groups. This resulted in our pups being brown and healthy adults, not prone to cancer and diabetes like the mother. This experiment proves that nutrition while pregnant is important in the health of their offspring, and the addition of methyl groups to their diet prevented various birth defects. With all of this being said, we can really change how our DNA expresses certain genes. Through our knowledge of disease and nutritional remedies, we may be able to live longer, healthier lives.